Bienvenidos al Seminario Polinterpares 2023. En un momento continuamos.
El Seminario Polinterpares 2023, Investigación y Publicación Impulsando la Ciencia, se complace en darles la más cordial bienvenida al segundo día de actividades. Les recordamos que durante la transmisión en vivo por el Facebook de Dirección de Investigación se estará transmitiendo QR para que ustedes puedan registrar su asistencia. El día de hoy arrancamos con una invitada especial, con el pie derecho, y es que vamos a iniciar con la conferencia magistral titulada Use of Systems Immunology for HIV Vaccine Development and Therapeutic Strategies, impartido por la doctora Isabela Pedrosa Pacheco. Para ello nos complace su presencia y es necesario conocer un poco más de su trayectoria. La doctora Pedrosa Pacheco es miembro del programa de desarrollo profesional en el Centro Crook Oxford Center de Inmunología, en el Departamento de Medicina de Notfield de la Universidad de Oxford. Obtuvo su formación en bioingeniería e inmunología en el Instituto Tecnológico de Monterrey y el University College London. En el 2014 se unió al consorcio para la vacuna contra el SIDA como becaria postdoctoral en la Universidad de Oxford. La doble experiencia de la doctora Pedrosa Pacheco en ingeniería y inmunología le ha permitido combinar un enfoque sistemático para comprender enfermedades complejas en un contexto integrador. La doctora Pedrosa ha realizado importantes contribuciones en el campo del VIH y la vacunología, desde revelar mecanismos celulares que regulan las respuestas de las células B foliculares y T colaboradoras, hasta descubrir características celulares predictivas necesarias en las estrategias de vacunación para desarrollar anticuerpos neutralizantes amplios y potentes. Mediante la aplicación de tecnologías novedosas y el desarrollo de metodologías que evalúan exhaustivamente las variaciones genéticas y proteicas que dan forma a la función celular. El objetivo general del laboratorio de la doctora Pedrosa es mejorar la comprensión de los mecanismos celulares y moleculares que subyacen a la regulación de las respuestas del centro germinal en la vacunación, la infección y el cáncer, para analizar objetivos que podrían usarse para mejorar las vacunas y las estrategias terapéuticas. Doctora Isabel, nos complace con su presencia, le cedo el uso de la palabra y sea bienvenida, por supuesto, aquí a Polinpares 2023.
Uno, dos, tres. Thank you, Aida, for the very kind introduction. It's a pleasure to be here, and in the spirit of publications in high impact factors, we thought it would be very beneficial to actually do the presentation in English and talk about the research and how we can make most out of our data. So today, as Aida mentioned, I'm going to talk about the research that we do in the University of Oxford, especially on how we use systems immunology for the development of HIV vaccines and also therapeutic strategies in the context of cancer, and how we have developed the methodology and technology and have made available data sets that then we can further mine to develop hypotheses. All right, so I lead the laboratory for functional immunology, which aims to understand the cellular and molecular mechanisms that regulate antibody responses. We are specifically interested in a process that is called the germinal center reaction. Now, germinal centers are temporary lymphoid structures that form upon vaccination or infection in our lymphoid tissues, or here in the ganglios. We would call it the ganglios. That are very important structures where B cells differentiate, undergo somatic hypermutation, and develop high affinity antibodies. Now, given the very important task of these structures and how specific this needs to be, these structures are tightly regulated by all cellular mechanisms around these B cells. Others have shown to the years that there's CD40 follicular helper cells, you can see here, that provide the cognate ligand interactions, the right signals to the B cells to undergo somatic hypermutation and develop these very potent antibodies. But we have also shown that there's other flavors of cell subsets, like natural killer cells here, CD4 Tregs, inside and outside these germinal centers, that can dampen or control specific responses. So altogether, all these cell subsets orchestrate to have a very specific response towards vaccination and infection. Now, I structured the presentation in three main things. So first I'm going to talk about the work that we did in the last 10 years as part of the HIV-CHAVD consortium in the US that looks at the immune regulation that underpins these broadly neutralizing antibodies in HIV infection. Then I'm going to move on to how we apply this methodology in the context of cancer and cancer vaccines. I'm going to talk specifically about the two different cell subsets that are important in these specific structures. And then how we can use and further mine these public data sets that we have generated to be able to cross-validate our data and also your data as well when you're thinking about the project, and being able to start strategizing to public in impact factor journals. Now, going into HIV. So HIV has been there for the last 30 years, and we haven't been able to develop a very efficacious vaccine as opposed to what we efficiently did with SARS-CoV-2 or with flu, with malaria, for instance. Now, why is it so difficult to induce broadly neutralizing antibodies, these very potent antibodies to HIV, and why is it difficult to develop these vaccines? And the answer comes in the structure of the HIV glycoprotein that you can see here on the right-hand side. You're probably very familiar to this type of structures when we look at SARS-CoV-2. And we know that in order for these antibodies to develop neutralization breadth, they need to target very precise conserved regions in this glycoprotein that is expressed in the HIV virus. And these are all colored in different flavors. And now the problem about these antibodies is that they see partial homology to self proteins that we have in the system. And this means that the B cells that are able to produce these antibodies, that are the precursors, are present at a very low frequency because they're often deleted by tolerance mechanisms. And this is the reason why we actually, when we develop a vaccine, then the B cells are not there to develop these broadly neutralizing antibodies. Now, we also know the characteristics of these particular antibodies. As you can see here, the structure, they have very elongated structures on the CDR, we call it the CDR-H3 regions, and they have features that really tells us that they rely on these structures that I explained in the previous slide. They really need to go into these germinal centers, interact with all the CD40 to be able to have maturation. With this, okay, so it's not only about the viral component, it's the immune system that is applying this pressure for these particular BNAbs or broadly neutralizing antibodies. So there is actually what we know in terms of the development of an antibody. In the first weeks after HIV, individuals start developing these non-neutralizing antibodies. And then after months, the virus starts developing mechanisms of evasion, and these antibodies are no longer favorable. And not only after 2 or 3 years of infection, there's a very rare subset of individuals that are able to actually develop these broadly neutralizing antibodies. A very excellent effect. And we were really interested in looking at these rare effects to see what's different in these particular participants that allows them to develop these broadly neutralizing antibodies. So to do this, we actually had access to a cohort of around 240 chronic HIV participants from whom we knew neutralization data. So what we wanted to understand is the cellular and molecular mechanisms of host control of these particular antibodies in the context of HIV, but also that apply systems immunology and mathematical approaches to determine what are the specific cellular features that contribute to the development of these antibodies. So the cohort, what we decided to do, so we screened these 239 individuals, and we decided to focus on the extreme ends of the cohort. So 50 individuals with the highest neutralization breadth, and compare them with the individuals with no or very low neutralization breadth. And we applied multi-omics approaches in the last 10 years. So we acquired data from these participants on the viral and host factors, so we knew which clade of HIV these participants had, also viral load, age, all the demographics, if they had cytomegalovirus serostatus, all the genotypic and genetic analysis as well. Then we did transcriptional profile. We from obtaining peripheral blood cells from these participants, and then we did whole transcription sequencing, and we were, you cannot see it very well because the contrast is not great, but we had gene expression of all these, of 20,000 genes across all these participants, and we started mining the data. As you can see here, I wrote R. So these are pipelines that we developed to actually mine the data from the gene level, and then pathway analysis to start understanding biological differences in terms of pathways downstream in terms of function. And then we can see that, and we have some blood transcriptional models that we know have been associated with a superior humoral response in the context of vaccination. We then had, with our collaborators in Stanford, we did all global antibody repertoire, so we knew about that these antibodies, the clonal expansion, what they were looking for, and we could actually dissect that these effects were systematic instead of very specific rare events. And finally, we developed methodology to probe the whole cellular immune environment, so we had a deep phenotyping in the cellular level. We developed computational pipelines to mine the data, biased and unbiased, and then we developed a lot of in vitro lymphocyte functional assays and cytokine profile. So we actually were able to accumulate to the years more than 8,000 features from these particular participants. Now, this really allows us to have a 360-degree information of the landscape, of the immune landscape of these particular individuals. And as a summary of all this data, we actually see that in the germinal centers, by looking at the blood, we see that these particular participants who develop protein neutralizing antibodies have global alterations in the IgG repertoire, higher prevalence of autoantibodies that seem to be correlated with the autoreactive responses. We have a higher response and a higher activation of the CD40 follicular helper cells that provide the right interactions for these B cells to develop these antibodies. And then in the context of regulation, we saw that every single cell subset that we measured, it was actually reduced in function. And so we see reduced function of the natural killer cells, Treg cells, and also regulatory cells. That is the paper that we're about to publish. And as an overall picture, we actually could describe that these particular participants seem to have an altered balance between CD40 follicular helper cells and regulatory cell activity that promotes the development of these protein neutralizing antibodies after 2 or 3 years of HIV. Now, at this point, we realized that we were capable to start applying mathematical models to determine if we could see predictors of protein neutralizing antibody response. So to do this, we ran around 6 to 7 machine learning models. And we were very surprised that despite the sample size that we had, because for machine learning models, as you know, you need thousands and thousands of data points, but also participants, we were seeing the same features coming through. And this really globalized in terms of we knew that these particular participants seemed to have a higher innate activation, they had a higher CD4 T cell activation as well, and also a reduced regulation, as I showed in the previous slide, that is really permissive of these protein neutralizing antibodies. And when we actually run the unbiased analysis, we were very happy to see that the groups really separated across the spectrum, obtaining all these 13 immune features. Now, the next thing to do when one does systems immunology is cross-validation. Is the most important thing. So we have done this in a particular cohort of participants that come from regions of sub-Saharan Africa, but then we started collaborating with other groups to actually cross-validate this in other regions. That's what we're doing at the moment. We're also cross-validating across species, so we have a model in non-human primates, and we're starting to see as well, I'm going to show you in the next slides, how this is, we're seeing the same similarities with the humans. And finally, the question that we will always get asked, okay, you have a snapshot of immune features after 2 or 3 years of infection, but when are these features coming? Are these features before HIV, or are these features that are actually accentuated or present in the acute phase of HIV, and how the dynamics actually come through? So for the non-human primate, for instance, what we do, we have a very interesting model
of infection. And this is really, really similar to what we see in the humans. Around 10 to 15% of the macaques actually develop broadly neutralizing antibodies. So we thought this is actually perfect to really understand the immune landscape and how similar it is. And so we can see as well, so we had the bulk RNA-seq data of total peripheral blood samples, transcriptional profile, and we developed a specific flow-based analysis and markers that were associated in the context of NHP that we can actually probe and compare. And what we started seeing is a very kind of difficult slide, but we can start seeing that actually half of the signature was, we were able to replicate it in these NHPs. And when we actually compare the models, we can see it's not perfect because of the cross species, but we start seeing some trends into separation of these particular macaques that develop antibodies or not. And, but one thing that was really striking when we looked at this particular model was that we started seeing the shared features, but we also started seeing unique features in the macaques that develop BNAbs. And this is something that we started comparing. So you can see this differential gene expression of individuals, humans that develop or not broadly neutralizing antibodies. And you can see there's thousands of genes that are different. This is slightly different in the macaques. In macaques, you see around 10 to 20 different genes that are actually upregulated. And we're starting to mine this data as well to actually cross-validate between species. Now, the question about longitudinal and dynamics of this immune landscape, we were trusted with these samples that were collected 30 years ago when actually the HIV infection started. And these were participants that were actually followed longitudinally, and some of them, unfortunately, acquired HIV. But this really allowed us to understand if these particular immune features were there before infection. And so you can see here on the left-hand side, this is our cohort of cross-sectional chronic infection after 2 or 3 years. And then this is a cohort of the Amsterdam cohort that we were entrusted with. So then we only had one vial of cells available, and we thought we need to make the most out of it. So we decided to actually shift into a more sophisticated instrument that was able to really profile these immune features. And we developed panels, around 5 panels that looked specifically at the cell subsets that are very important in the context of germinal center reaction. And we also included high dimensional plasma analysis as well that we did in Oxford to have most of the information from these particular patients. And just as a summary, so you can see, for instance, the cellular features on the right-hand side. These are all features that are starting coming up that are different in these particular participants in the chronic phase, so after 2 or 3 years after HIV. Also, all these features in plasma. And when we compare this with our signature, we also started seeing as well that there are similarities between our Amsterdam cohort and our Chavdi cohort. All right, and you can see here in this plot very nicely how the peaks indicate if there's a differential expression between the individuals that develop these antibodies or not, and you can really see that most of the peaks are going up, so everything is going in the right direction in these particular landscapes. Okay, then we decided then we actually went back and say, okay, before HIV, can we see these features? And the answer is yes. So you can see all these blue features coming through, as well in the plasma analytes. Things are going in the right direction. And all these peaks are really indicative that we're starting even seeing this information before infection, which suggests that there might be a predisposition for these individuals to develop these antibodies. All right. And then finally, we actually looked in the very acute phase of infection, so in the first months after these participants acquired HIV, and we started actually seeing that all these features were actually accentuated in the very first months, and then settled in the context of chronic infection. So really, this longitudinal cohort allows us to have a profile on the dynamics of these particular subsets, and that everything is really going in the right direction even before infection. Okay, so as a summary of what I show in terms of HIV, we actually were able to develop systems immunology approaches that revealed a particular signature that classifies broadly neutralizing antibodies in the context of chronic HIV with high accuracy. We identify parallels between the non-human primates and the humans as well. Then we were the participants who developed these broadly neutralizing antibodies seem to have a poised environment that favors this, the development of these BNAbs in the context of infection. And finally, this suggests that in terms of vaccines, that we need to actually develop vaccines that not only look at the immunogen in the context of the virus, but also develop and think about smart adjuvants that actually promote this particular immune environment that can facilitate these broadly neutralizing antibodies in the future. So in the future, we know that it's important to actually do big data and apply machine learning models and systems immunology to do much better vaccines in that context. Now moving into cancer. So we spent 10 years doing these methodologies, trying to really probing the immune landscapes. And so we thought it was a very good opportunity to move into the cancer context. Now, very interestingly, in the last 10 years, we clinicians around the world started observing these particularly lymphoid aggregates that seem to be associated with longer overall survival or better response to treatments. And these structures seem to resemble what we were actually seeing in the secondary lymphoid organs that we have been discussing in the last 20 minutes. So these structures are they were starting appearing, for instance, across all cancer types. So we started seeing them in ovarian. Then we saw it in melanoma. And then it went across colon cancer as well, lung cancer. And people started to become very interested about these particular structures from the perspective of therapeutic strategies. Now, as I mentioned, with very few exceptions, these structures are associated with a better prognosis in the patients, but we don't know much about them. So we don't know what are the drivers of these structures and what is the contribution towards anti-tumor responses in that context. So you can see here in the figure that this is how these lymphoid aggregates look like. And so it's very important as a scientific community to actually dissect the cellular composition, the spatial distribution, what are they seeing, so the antigen specificity and the cellular function. So we focus specifically in colorectal cancer because of two different reasons. The first one is the clinical perspective. So as we have around almost 2 million new cases reported in colon cancer worldwide, and the survival rates really depend on the timing in which the patients are diagnosed. So for instance, when you are diagnosed in stage 3, you can the overall survival is 4 to 6 years. So very interestingly, in early stage patients, these tertiary lymphoid structures seem to be associated also with better prognosis and after resection, suggesting an immune-derived benefit. And there's small CRC cohorts that suggest that if you have these very organized structures, then you actually do much better as a patient. Now, we took the approach in the context of from the HIV perspective, and what we did, we started actually mining public data sets from TCGA. And what we did is we knew there were specific cell subsets that were very important for this particular germinal center reactions. So what we did is basically take the data that is publicly available from 181 patients, and then stratify them in between upper and lower quartiles of specific responses. And then we applied mathematical algorithms to actually estimate the cellular proportions from bulk RNA-seq data. And we were able to start seeing specific differences in longer overall survival. So these are the Kaplan-Meier curves. So in the x-axis, you see time of survival, and in the y-axis, you see the percentage of patients that are being kind of analyzed in these particular curves. And you can see when you look at the CD4 T cells, which are, as I mentioned, are very important for the germinal center reactions, there doesn't seem to be a difference in survival between the individuals who have more CD4 T cells, as shown in red, and the ones that don't have the T cells, as shown in blue. But then when we started looking at the very specific functional subsets that look at the germinal center we do start seeing a separation between the two groups that is still not significant, but there is a trend. And in the same way, when we look at the regulatory CD4 T cells, is actually shifted. So this was highly reminiscent of what we were seeing in the context of HIV. And when we look at the B cells as well, which are the most important cells that produce these antibodies, you can see that the individuals that have more B cells, estimated B cells, they seem to survive longer than individuals that don't. All right. So as I mentioned at this point, so we had access to tissue biopsies from these particular patients. And one of the challenges about this is that the sample numbers are extremely low. So we needed to apply the same methodology from the HIV and make these panels as comprehensive as possible to get most out of these particular cells. So what we did, we used a spectral flow cytometry and actually collaborated with BioRad, which are in these very specific probes with a specific spectra that allows us to actually put as many colors as possible in one particular panel. And we were able to actually develop a 38 color panel that is able to identify almost 70 populations and resolve 20 distinct immune clusters. You can see we then apply computational pipelines, both biased and unbiased, that we have set up in the lab. And you can see here all the markers that are included in this particular panel. And then it's data-driven approach. So you put all the data and then using cluster algorithms, then you start resolving this particular immune cells subset. So you can see we have around 20 different immune clusters. And when you look at them in the principal component, the TISNI panel, you can really see the different immune cell subsets with different functions. You can see the B cells in here, the NK cells, the monocytes, CD4 T cells, CD8s, gamma delta, all the immune cells that are important in the context of the germinal center reaction. So we were very happy with this methodology and the technology that is provided by the spectral flow cytometry. And so we decided to develop 5 panels with this that allow us to sample almost 150 proteins at a time. So with this, then one of our students she set up all the panels and QC them as well. And then we started with a very pilot study with 3 samples that came from patients that were diagnosed with stage 3 and that were scheduled with resection. So we took the biopsies and together with the Oxford Cancer Consortium, we actually did multi-omics approach. So we have single-cell RNA-seq data and all the multiplex analysis to be able to pull all the data together and start doing systems immunology approaches. So the first level information that we got was, and the goal was to actually, can we characterize the main subsets in these particular biopsies? And the answer is yes. We were able to actually characterize CD3 T cells, all the flavors of T cells, as you can see here, and the B cells you can see here. There's a lot of donor-to-donor variability, which is very important and relevant in the context of function that we thought that was very interesting too. And you can see it very clearly here that depending on the patients with, for instance, different types of mutations that are being diagnosed, you can see the immune infiltrates really range from 3% all the way to 20%. But when you look at the flavors of the immune cells that are able to migrate near the tumors, you can see that, for instance, the CD4 T cells are quite similar across the board. B cells really are different as well. So that really shows that there can be an immune benefit that could be harnessed and can be specific for a patient. Now, so zooming in into the CD4 T cells and applying our computational pipelines, in here you can see every dot represents a cell. So we pull all the data from the biopsies, the tumor biopsies, and then we compare it to adjacent normal tissue as a control. And you can see that we were able to resolve 12 different metaclusters with different colors in here. One metacluster we were very interested about is actually here in the corner, which seemed to have characteristics of T follicular helper cells that, as I mentioned, are very important to help B cells to develop antibodies. And most strikingly, this particular population was not present in the normal adjacent tissue, which tells us that there's a signal that is triggering these particular cell subsets into the tumor biopsies. We then decided to actually follow that path and get more information of that particular metacluster. So we then characterized it with the flow cytometry. You can see based on the expression of these two markers, you can see a very nice population in the biopsies as a comparison to what we normally are expecting to see in the secondary lymphoid organs. And we can characterize up to 10% of cells within the CD4 T cell compartment that seem to have the potential to help B cells. And you can see it here with the different flavors. Now, we then did apply data-driven approach, and we asked, okay, can we actually look for a unique feature or unique cluster within this particular population? And we were very happy to see this unique meta-cluster, again, with the highly activated features that really gives us insight into the functional capacity. So then and this seems to be actually very unique in the tumor biopsies and not present in the normal healthy adjacent tissue in these particular patients. Then what we did is, okay, let's mine this in the public data sets because we're always facing the challenge. We knew there was a pilot study. We only had 3 particular patients, but then we can really utilize what's available in the CGA, which is a data set by the NIH. So we took as a particular signature that characterizes these T follicular helper cells, and we actually found that when we looked at the colon cancer patients, there was only one subset of patients with very specific features that seemed to be associated with longer overall survival. So if you had this TFH, your survival was much longer and significant as a comparison to individuals who didn't have this particular signature. Now, as I mentioned, at this point, we knew that the phenotype, so we could characterize this particular subset, but it's very important in cellular immunology to actually validate in a functional capacity. Now, T follicular helper cells are able to secrete a cytokine which is called IL-21 that is very unique for these features, and that is very important for help towards the B cells. So we developed these functional assays, and we can see as well that in the CRC biopsies, there seems to be an expansion or an enrichment of these particular subsets secreting IL-21. And you can see here that is very unique as a comparison to the other flavors that we were seeing in the CD4 T cell compartment. And in the same manner as what we did in the phenotype analysis, we actually could see that in this particular subset of patients, having these particular cells seem to be associated with longer overall survival. Now, so I talked about the CD4 follicular helper cells, and then the hypothesis is that then the B cells are going to be associated as well. So then we look at the B cells, and as a comparison to the CD4 TFH, we were only seeing around 0.2 to 1% of cells in this particular biopsy. So these cells are very rare. However, we could see different flavors of these particular cells, and most of them were actually from the memory compartment, which suggests that these cells have seen antigen and they're specific to a particular response. And these were the ones that were near the tumors. However, when we plugged the data into TCGA, we were very surprised that the memory B cell compartment was actually the other way around. If you have more memory B cells, you didn't seem to do better in terms of prognosis. Then we actually thought, okay, maybe it has to do with a particular B cell subset that has already acquired this help from the CD4 TFH and is in the last state of differentiation. And these are called the plasma cells. So these are very professional cells that secrete a lot of cytokines that are really important for instance, vaccination. And we could actually characterize the cells in the biopsies with a higher enrichment of IgS cells that is not unexpected because this is gut microenvironment. And when we did the data-driven approach, we were actually focused on this particular unique meta-cluster with high activating features. So then we did exactly the same exercise, go back to the public datasets and ask if this particular signature is associated with longer overall survival, and the answer was yes. So in these particular subsets, if you had more plasma cells, you seem to do better. And when we looked at the correlation between the TFH signature and the B cell signature, making a lot of sense biologically speaking, you could see almost a perfect correlation between the two signatures, not only in the tumor tissue, but also in the healthy normal condition, which suggests if you have a B cell or a plasma cell, you have a CD4 TFH nearby. Now, I talked a lot about characterization, but in the beginning of the talk, I actually started mentioning the importance of the structure and the location of these particular features or these particular cell subsets. So then what we did is actually apply these particular cell subsets into the spatial context. And to do this, we again mine a public data set. This is actually a data set from spatial transcriptomics from the Illumina company, which is available. And this is a tissue section with the specific dots that actually accumulate 1 to 5 cells, and you have all the transcriptional profile from every single spot. So you get something, you get your H&E section, which is a 7mm section in here, and then you can overlay all your transcription profile. I don't know if you can appreciate the dots in here in the zoom-in section. Now what we did, we basically, based on the transcriptional profile, we actually color-coded the plasma cells as shown here in blue, and then looked at the T follicular helper spots. And you can see here in the corner that there seems to be an enrichment of both CD4 TFH and B cells that seem to be suggestive of the presence of a tertiary lymphoid structure. But we wanted to have a more quantitative approach towards this, so we actually team up with the mathematicians and physicists at in Oxford, and we actually did an analysis to look at the patterns of proximity between one cell towards the other. And what we actually could appreciate in here is that whatever is in this box seems to be clusters. So we could see that the B cells are shown in red. They seem to be clustered within each other and having a pattern in the first radius of this spatial location. But the TFH seem to be dispersed across. So this really told us, or we're suggesting that, for instance, the B cells they're like to be together to actually kind of form or they're perhaps responding to a particular antigen that is coming from a cancer type. TFH cells are clustered within the B cells to provide the help, but they're also dispersed across the tissue, what we think is perhaps patrolling. And this is something that is reminiscent of what we see in the secondary lymphoid organs upon vaccination and infection. So as a summary, I show that we have developed all multi-omics and functional platforms to be able to quantify the phenotypic and functional heterogeneity of these particular cell subsets in the tumor microenvironment. And this really allows us to develop hypotheses that these particular subsets provide an immune benefit in these patients. And we are in the process of doing further functional and characterization of these particular subsets as well, to be able to leverage in a therapeutic strategy for these particular subsets. And currently public data sets to be able to validate the biology. So what we think is happening in a particular tumor microenvironment is this help between these now going into the objective of the how we can actually mine data, how we can use it. So as you have seen in the presentation, big data requires coordination, and it requires a consortium. So these are assays that are often economically challenging to, so you need a lot of grants, you need a multidisciplinary team. And so there's huge consortiums that have provided efforts and investment into actually providing these public data sets for people to start mining it. And I'm gonna talk about 3 specific examples. So the first one in the vaccine research field, the NIH has curated in the context of the Human Immunology Project Consortium. And you can see this is actually a review that I wrote in Nature Reviews Immunology, that actually they were able to follow they were able to use different platforms integrating transcriptional data and cellular data of over 3,000 peripheral blood samples from 820 adults across 28 studies of 13 vaccines. And with this, the authors were able to normalize and made it publicly available in this website, Immunospace, to be able to dissect pre-existing and early transcriptional signatures associated with high antibody titers. And the vision of these particular data sets is that people that comes with different, for instance, data from different geographical locations are able to actually cross-validate with their studies. It's freely accessible. The other big data that I think has really revolutionized the way how we do science in the context of cancer is the Cancer Genome Atlas program, also funded by the NIH. And this data set integrates 20,000 primary cancer and animal samples spanning 33 cancer types. It's a joint effort between NCI and the NIH-GRI that began in 2006. And TCGA, which is actually the data set that I mined across in the presentation that you saw, generated over 2,000 petabytes of genomic, epigenomic, transcriptomics, and proteomics data. And so this is a very beautiful resource as well if one has a specific hypothesis and we don't have enough and we don't have the data. Just go there and look for that. And the last one, for instance, is the aspect that big data is deposited in repositories as part of a publication requirement. And you can see, if you look, for instance, in all these new publications, you are always going to see big data being available. And this is one example, for instance, what I showed in the HIV field. We generated bulk RNA-seq data from HIV patients, and we actually deposited all this in GEO, which is the NCBI data set. And so we have, of course, generated hypotheses. We have certain findings, but then we wanted to ask, like, how we can further mine this data. And one of the questions we had and what we saw in this particular cohort is that these particular participants, the immune landscape seemed to be reminiscent of immunity. And that actually makes sense in the context when you look at the antibodies. So then we wanted to ask what are the shared principles of immune dysregulation in these particular participants as a comparison, for instance, to a lupus patient. And to do this, for instance, we have a transcriptional profile, but we didn't have the transcriptional profile of the lupus. We wanted to generate hypotheses. And to do this, we actually looked for all the public sets of bulk RNA-seq data and found a cohort that was actually feasible to actually normalize and look for it. And we also had all the immunological environment as well, so we can generate and validate the hypothesis in the functional level. So this is just a quick peek of what we have done when we compare the two subsets without spending any money. So we, in here you can see the UMAP analysis. Every single dot represents a participant. And you can see nicely that the lupus patients clustered together, and these are all the HIV participants. And we then did, we mined the data thoroughly, so we looked at the genes, at the pathways, at the blood models, and what we were very interested in, it was the unique and shared features between the HIV participants who developed the antibodies and the autobody patients. And that really provided information on specific pathways that seem to be associated and that could be relevant in the context of vaccine strategies that we're further mining. As you can see here, this is all kind of proof of principle. So I show you like all the process on how to generate data, but also how to cross-validate your results without having to spend, invest in the bright grants, et cetera, et cetera. And this is something that is important to have clear from the beginning. What is the objective when one starts thinking about using public data sets for a publication? And as an editor of the Nature Communications Biology, I can give you some tips of what we're looking for. So key questions to look for when one is planning a manuscript. So what is new here? And where do you want to go? Is there an advance over the current literature on the topic? And most importantly, if yes, is this interesting for researchers in the field and how we can contribute to the scientific community? The next step is actually to check in the literature. Go to PubMed, go to Google, use all the resources to see, has anyone already reported this? Because then it is a waste of resources in time as well to be kind of doing research that is already kind of out there. Target your journal. Look at the journal that you are aiming for and then ask, are there similar papers published in the journal? What is their approach? What is the introduction? What is their aim? And then structure your manuscript. Or start organizing the figures, check similar figures in the target journal. Presentation is a key thing, right? So if you polish the data, as you know exactly what journals are looking for, it is more likely that you're gonna be, you get a positive result from the reviewers. Ask for help. I think this will be what my top tip. Manuscript, review the manuscript, send it across your colleagues. If you have a scientific writer who is available to you to check the structure, to check the grammar, the flow, use those resources, because when one comes as an editor, you see a manuscript, you really need, it's like a pitch elevator, right? It needs to be presentable, it needs to flow, it needs to be concise, the structure needs to be there, and that will increase the chances of success. Be resilient and open to reviewers' comments. So reviewers can often be quite aggressive or not aggressive, but then it is always the peer review process, and this objective of this talk is to improve your manuscript. So dissect, take the time to actually understand where they're coming from, what they're looking for, and if this is feasible and how to approach it. Iterate and improve, and don't give up, especially. So we're used to actually have several iterations of manuscripts. The more that you actually work on this manuscript, the more likely you're also going to get accepted, and the better the research and the data that you can provide to the scientific community. And so with that, I just like to acknowledge all the teams and the people all across the world who has been involved in particular studies in the US, in Amsterdam, in the UK, and also in Mexico. And you for your attention, and I would like to open the floor for any questions and actually discussion on how to publish these papers and how to organize them. Thank you. Right.
Muchas gracias, doctora. Pues aprovechemos este espacio para abrirlo a preguntas y respuestas, alguna duda o algún comentario que pudieran tener los alumnos aquí presentes o a través en línea. Si gustan alzar su mano, les pasamos el micrófono, y si no, creo que por el momento no. ¿Sí? Perfecto, entonces le pasamos de este lado el micrófono. Si nos da su nombre, y con mucho gusto le cedo el micrófono a la doctora para que conteste la pregunta.
Hi, I'm Diego, and I have two main questions. You mentioned that you use some machine learning algorithms, but which of those models works the better for your research? Because I understood that you were doing some finding the important features, but it was just like a feature selection, or what kind of algorithms did you use? The second one is, if during your research, had you ever used any deep learning architectures? Because I mean, you have tons of data, so probably it works better, I don't know.
Yeah, that's a very good question, thank you for that. We decided that the approach, given that we only had 100 samples, we had 8,000 features from 100 samples, to use the very simple regression model. So as a machine learning, that was the first. That actually gave us the best in terms of feature selection. It's first comparison of what is significant between the two groups, and then we ran the features that were the most models from that. So when I talked actually about 6 models, yeah, we started from the very simplistic, where we actually needed to compromise on the sample size, because as you know, data scarcity is the main problem for systems immunology. So we needed to say, okay, we have a small sample of participants with one model. And then we did subsets from there to bring in the biology in the models. Because one thing is, and as we all know, if if you give data to the AI, it will give you data. It doesn't mean that it's biologically relevant. So we felt that it was important to start iterating from the biology perspective and ask, okay, we knew the end cases were important. Let's put them back and let's ask if the model improves. And it was. So we started kind of moving the model and iterating further and further and further. And the last one we actually did, we ran models to account for data scarcity. And then we say, okay, what are the hits of these features that keep on coming, and then we actually validated with the functional assays. So that was the port. In terms of kind of more sophisticated models, we felt that we were underpowered on that sense. But you can do this with all the genomic data that you have available and all these public resources. Like if you have a very specific resource, that would be perfect. And it's actually the way forward in terms of medical sciences. So we have it's going to revolutionize, it's going to help as well the teams to make decisions in the future. Yeah. Any questions about publications or structures, or is anyone planning a manuscript in the future? No? Yeah. Okay, well, with that then, I'll pass it.
Digo, la oportunidad está abierta a que hagan la pregunta en español y la doctora puede contestar en cualquiera de los dos idiomas. Si no hay alguna duda por el momento, pues agradecemos mucho su participación. Si gusta acompañarme de este lado para hacerle entrega formal de esta constancia. Y que, bueno, dice al pie de la letra, el Instituto Politécnico Nacional, a través de la Secretaría de Investigación y Posgrado, otorga la presente constancia a la doctora Isabela Pedrosa Pacheco por su participación como ponente de la conferencia magistral Use of Systems Immunology for HIV Vaccine Development and Therapeutic Strategies, pues realizada en formato presencial el día de hoy, 16 de noviembre del año en curso, en el marco del Seminario Polinterpares 2023, y firma la doctora Ana Lilia Coria Páez, secretaria de Investigación y Posgrado. Doctora, muchísimas gracias. Le hacemos entrega también de este pequeño detalle por parte de la Secretaría de Investigación, ¿no? Y bueno, un fuerte aplauso, por supuesto, a la participación de la doctora. Muchísimas gracias. Esperamos que pueda venir nuevamente a cubrir un espacio como lo es Interpares 2023. Bueno, pues con esto estamos despidiendo prácticamente a la doctora Isabela Pedrosa Pacheco y agradecemos infinitamente su presencia y hacemos una pequeña pausa, no sin antes recordarles que en unos minutos más daremos inicio a 10 consejos para publicar en revistas arbitradas por el licenciado Sabino Seguro en Polinterpares 2023. Contamos con la presencia de la secretaria. Se gusta subir aquí para tomarse también la foto con nuestra ponente. Ya le dimos la constancia, pero recordarles, por supuesto, que ustedes están aquí en Polinterpares 2023. El seminario busca reunir a la comunidad científica, profesores, estudiantes de licenciatura y posgrado, así como editores, autores académicos y público interesado en el tema. Les agradecemos, hacemos una pequeña pausa y regresamos en breve a Polinterpares 2023.